Howdy, folks. Thanks for checking in to MrUlrich'sLandofBiology.com. I'm Mr. Ulrich, and this is the inner sanctum of the office at Upper Twangville, the home of Mr. Ulrich's Land of Biology. In this notescast, we're going to be looking at and talking about macromolecules, those larger molecules that make up living things. They are created by taking the smaller molecule subunits, much like those functional groups that we were talking about in previous videos or the other day in class. These larger organic molecules are really amalgamations of those functional groups. Now, I am showing you these donuts for a reason. If I were to feed you only donuts, it's not as if you would turn into a donut. It's just that those compounds that you need to make all the lipids and the carbohydrates and the proteins and the nucleic acids that make up your cells and yourself would only come from those donuts. So you'd be limited to donut lipids and donut carbohydrates and donut nucleic acids as your raw materials. If we're going to discuss macromolecules, we really need to take a moment to talk about polymers, which are kind of a basic structure that is common to many macromolecules. Polymers, just like the prefix poly indicates, are many. They're made of chains of many repeating units in a row. I often compare them to choo-choo trains, rather simplistic, but it's kind of cute. That means that one car of the train would be a monomer, mono for one, and two cars would be a dimer, and three cars would be a trimer. And there are oligomers, which are several, and when we start talking about protein structure, we can talk about hexamers and things like that of insulin. Sometimes those are not in a chain, however. The important thing is that these chains are bonded together covalently, so that there are covalent bonds between the monomers. Speaking of those covalent bonds, how do we actually make those bonds, those chains of repeating units? These are going to be condensation reactions, usually between a growing polymer and an adding monomer that's being added on. In the case of living things, it involves taking a water molecule out. One side of the growing chain provides the hydroxy, the other provides a hydrogen group. Those together make the water. Because we're removing water to put things together, it is literally dehydration synthesis. Now, this is not going to take place on its own. It would take more energy in order for these molecules to just clunk together with just the right trajectory, just the right amount of energy to knock the hydrogen and the hydroxyl group off. So it's not going to happen on its own, but if we add things that will hold the subunits in place at just the right angles and put just the right bond strain on those angle strain on those bonds, we can break the bonds a lot easier, and this is going to be done through enzymes. When you go to your favorite restaurant that isn't a fast food place, one of the earlier questions you're going to get is, do you want a glass of water with your meal? Do you want water with that? They're probably not thinking about the chemistry of digestion, but since digestion is going to involve breaking those bonds that we were just talking about forming during dehydration synthesis. The process of digestion involves hydrolysis, which is just that, the reverse of dehydration synthesis. It's taking water molecule, breaking that into the hydrogen and the hydroxyl, and using those to kind of stuff into that bond between the two parts of the polymer and break that apart into now two separate molecules. Sometimes this even uses the same enzyme as dehydration synthesis in a reversible reaction. So let's start talking about some macromolecules. The first group we'll talk about are carbohydrates. Later on, we'll talk about lipids. I'll probably save proteins and nucleic acids for another video. But carbohydrates here, we're talking about both the complex carbohydrates and starches as well as simple sugars. Just to run through the basics here, carbohydrates are compounds that are made of carbon, hydrogen, oxygen. The term carbohydrate does indicate something about their structure. The carbo comes from the carbon, and the hydrate indicates the same ratio hydrogen to oxygen in a carbohydrate that you're going to see in water, hydrate for water. There's good old C6H12O6, which is glucose or fructose or galactose. We'll talk about isomers later. The important thing here is that glucose is the primary fuel source for all living things. So there, as far as carbohydrates go, an important function is energy, not just as the raw fuel, but also because we can glue these molecules together and form starches, just like potatoes do for storing energy. And as well as start to make complex carbohydrates for cell walls or insect exoskeletons, the crunchy parts of insects. It's important also for us to note that the monomer, the repeating unit of complex carbohydrates, are simple sugars. The names of sugars are also important. Nomenclature is something we should be aware of. They do usually end in "-ose", so terms that end in "-ose" are indicating that it is a sugar. They are classified by the number of things in the ring. It doesn't have to be a carbon. Sometimes it's oxygen, as you can see in ribose there, and deoxyribose. Glucose is a hexose sugar because there are 6 members in the ring. Deoxyribose and ribose there, and fructose even are pentose sugars with 5 carbons. There are trioses. You'll notice that's not in a ring. It doesn't have to be in a ring structure, but that is a triose. They're glyceraldehyde, which we will discuss when we get into energy. Just a quick run through the functional groups in case you forgot or need to review from the last time. There are some functional groups that we should be familiar with, like the carbonyl. Aldehydes are compounds that have a carbonyl, where the carbon is attached to at least one organic compound, one carbon. Ketones are attached to 2. There are aldoses, the types. These are sugars that are open, that are not in the ring structure, where they'll have a hydroxyl group. It is also important for us to recognize that sugars will and can be found in their open and ring forms. Pure sugars are linear or ring forms, but when we start to dissolve them in aqueous solutions, they take on the ring structure. Since we're going to be talking about living things that are large aqueous solutions, we're always talking about the ring structure. We should know something about the way carbons are numbered, however. Carbons are numbered based on which carbon you are relative to the oxygen that is on the terminal carbonyl group in the aldose. For our purposes here, we're talking about this first carbon attached to the oxygen, going all the way around 1, 2, 3, 4, 5, and 6 prime. The 6 prime in glucose is the one that's hanging off the end. Just to describe them from simple to complex, we have monosaccharides. These are the simple sugars with 1 ring. Those come together to form disaccharides like sucrose, which are 2 rings. And then we can start adding those rings together and start to make polysaccharides, which are the polymers forming starches. To make a disaccharide, we are going to go, like I said before, through good old dehydration synthesis involving monosaccharides. Again, this is an enzymatic reaction, this here forming maltose, where 2 glucose molecules come together through dehydration synthesis. This bond that is formed is between the 2 simple sugars is referred to as a glycosidic linkage. To take a moment to talk a little more about those polymers of sugars, the polysaccharides. It's pretty simple. It takes not a whole lot of energy to start to bond those simple sugars together to make the complex carbohydrates. So these make for really good stores of energy. And both plants and animals do utilize this, taking excess amounts of sugar, simple sugar molecules, and gluing them together into starches like glycogen or amylose, amylopectin. Because of this ease at which you can make complex carbohydrates, or plants can, they also are used as building materials, making cell walls, and certainly we see this in animals as well, as arthropods use them to make the exoskeletons. I hinted before at amylose. We have been talking about these polymers as long chains, but they don't have to be linear. They can be branched. This is the difference between amylose, which is a simple starch, and amylopectin, which is a little more complex. We start to see different bonds between the simple sugars. And that forms a branching pattern. When we start to look at glycogen, we see that the branches have branches, and so it becomes very complex. This is very helpful because the molecules that are going to digest these starches to yield the simple sugars for energy start on the ends of the molecule, and so if you have lots of branches, that yields lots of ends, and it makes it really efficient to utilize glycogen as an energy source when necessary. Not all glucoses are the same. It's a little strange. When we start talking about the stereoisometry of glucose, the nuts and bolts of this are not something that'll be on the AP exam, although it is interesting and certainly kind of cool. This carbon right here is kind of special because it's attached to four different things. Here's a hydrogen, here's an oxygen, here's a carbon, here's a hydroxy group. Because it's attached to these four different things, it has a cool characteristic that's called stereoisometry. It has handedness, kind of like your left and your right hand they're the same thing, the thumbs, and they're all arranged, but they're not superimposable over one another. And so you can't put your left hand in your right hand glove. And that's important for us as biochemists because shape is going to be very important in order for enzymes to be able to complex with substrate, since that is a shape-shape interaction. When things start to have different shapes, then that starts to affect whether it's going to be able to be complexed with that particular enzyme. Love potatoes. Love them. Why do we like potatoes so much? Well, because potatoes store their excess glucose in alpha glucose molecules in a long chain in a form of starch that you and I have enzymes that we can break down and utilize. Now, cellulose is also a chain of glucose molecules, C6H12O6 attached to another C6H12O6 all the way on down the line. But the reason why you and I can't go out and eat, hay like a cow, is because we lack the enzymes. We don't have cellulase that we need in order to break down the different type of 1,4 glycosidic linkage between those beta glucose molecules. It changes the shape of the molecule enough that it doesn't complex with enzymes that you and I have. We do have enzymes that can complex with the alpha glucose monomers all in a chain, but not the beta glucose monomers in a chain. Cellulose, unfortunately for us, is like the most common compound out there, and it's too bad that we can't use it as an energy source because it's got a lot of energy in it. Diabetes is a condition where a person has difficulty maintaining a stable blood sugar. People who are insulin dependent need to be able to look at the food that they're eating and have an idea of how that's going to affect their blood sugar. So different foods are given what's called a glycemic index. The glycemic index is just that. It tells how quickly that food is going to raise the blood sugar. So of all these different compounds, these different foods, these different fuels, of all these different foods, which one's going to raise your blood sugar the most? Well, peanut M&Ms, candy, which you might think, oh wow, that's candy's going to raise it quickly, has one of the lower glycemic indexes. Natural fruits also pretty good as far as keeping your blood sugar relatively low. Once we get into complex carbohydrates like bagels, which are actually soaked in malt before if they're done right, uh, have quite a bit of sugar in them and quite a bit of carbohydrate. That's all they are. But really, the bad guys are the rice cakes and the corn flakes. What do you think about it? That's breakfast cereal. It's supposed to like pick you up and give you the energy that you need to go through the morning. This, of course, brings us to my favorite group of macromolecules, the fats. Oh, do I like the fats? Anyway, this also includes the oils and waxes, which I'm less interested in eating, although olive oil, of course, delicious. Fats, lipids. Let's get back to really what we should be talking about, which are lipids. Lipids are great long-term energy stores. Ounce per ounce, fats and lipids contain far more calories than carbohydrates, the next best energy store. It also is a structural component of cell membranes and all kinds of other stuff. So remember, you need fat. You're made of fat. Yeah. Okay, so lipid basics. Also, they are also composed, just like carbohydrates, of carbon, hydrogen, and oxygen. Difference is going to be that they have these great long chains of carbon and hydrogen, and just a few oxygens. So the ratio there of hydrogen to oxygen is going to be much different than that of water. We can talk about different kind of like family groups. These are loose classification groups like the fats and the phospholipids and the steroids. These are, like I said, different very loose classification groups that we use. It is important to note that we are not talking about polymers, even though they have those long hydrocarbon chains. We don't consider lipids carbons. They are big, big, big molecules, though. These are really, really huge and enormous. The fats that we're going to be spending most of our time talking about are fats that are made from one single glycerol molecule and then three fatty acids. Glycerol molecules are three carbon, one, two, and three carbon, what they're called triols, and that's because each carbon here has an OH group attached to it. The fatty acid itself has a carboxyl group on one end and then a big long chain of carbohydrates at the other end of it. They're named based on the number of carbons and the nature of the bonds between those carbons and stuff like that. Now those that are made this way, the three fatty acids with a glycerol are called triacylglycerides or triacylglycerol. It does set up an ester linkage between that hydroxyl of the glycerol and the carboxyl group of the fatty acid, and so we refer to that as an ester linkage because it has the central oxygen here between the two different carbon species, so it looks like an ester there. Looking at lipids more functionally, they are mostly hydrocarbon chains. When we talk about hydrocarbon chains, that's hydrogen and carbon are certainly nice coordinating covalent nonpolar bonds. But it does have that carbonyl group in there. But because so much of it is hydrocarbon in nature, the overall molecule is therefore hydrophobic. It doesn't mix well with water. Oil and water don't mix. So the function there, the primary function that we think of is energy storage. It's because it is a good concentrated store of energy with all of those stable hydrogen-carbon bonds. They'll yield quite a bit of energy when they are broken, when we strip those hydrogens off. They do have ounce per ounce, 2 times or more the caloric value of complex carbohydrates. But energy is not the only thing they're good for. They're also good for cushioning organs from blows and injuries. They are also good for insulating. Think whale blubber when we think of insulating against the cold, like cold water. It also creates a large thermal mass. One class of lipids we need to be familiar with are the saturated fats. Again, lipids get their characteristics largely from the length and nature of the fatty acid chains, the hydrocarbon chains from the fatty acids. In saturated fats, the carbons are all bonded to as many hydrogens or carbons as they possibly can. So there are no double bonds there between the carbons. That gives us straight chains. It allows those three fatty acid tails to kind of fit together, nice long and straight nest together. This is what we see in most animal fats like butter, and those that are solid at room temperature because they are more dense. They are able to fit together more molecules because those legs nest together so well. Unfortunately, though they are tasty, these are ones that do contribute to atherosclerosis, the building up of plaques on the inside of arteries, high blood pressure, and other kinds of fantastic cardiovascular disease. We should also be familiar with the unsaturated fats. Since the saturated fats have the straight chains, the unsaturated fats have the bent chains. This happens because there are carbon bonds that are double bonds rather than single bonds. Monounsaturated fats have one double bond. Polyunsaturated fats have several double bonds between them. These are the ones that are more common in plants and fish. They are liquid at room temperature. This is because those individual legs don't nest together so well because they're the bent chain, the bends at each one of those double bonds keeps them from nesting together as well. Another class of lipids that we should be familiar with are the phospholipids. These have a glycerol, but in two fatty acids. Instead of a third fatty acid, it has a phosphate group. That is a big negative charge group sitting up there. Because of this charged head on it, we call that the head region, is this hydrophobic or hydrophilic? And the answer is yes. The tails, the heads are, with the phosphate groups, are hydrophilic, very polar in nature. And the tails with all those nonpolar carbon-hydrogen bonds and carbon-carbon bonds are very hydrophobic, are very nonpolar. That means that the tops of the molecules mix well with water and the tails of the molecules do not mix well with water. And this nature is going to be real important when we start talking about the structure of living things and cell membranes. So what do these phospholipids do when I put a bunch of them in water? Well, since the heads of them like the water, they're attracted to the water. They're both nonpolar, so they get along. They don't have any problem being there. It's the tails that have the problem. The tails are nonpolar and they don't like being in the water. They're not attracted to water. They like being with other nonpolar compounds. And so where can they hide? Well, they can hide in the tails of other phospholipids. And so they'll spontaneously form these structures called micelles. Where the tails all are nested together, making a non-polar area on the inside, and the heads are all on the outside mixing with the water, which of course is polar. We can also put some water on the inside of this micelle, and we'll start to form this bilayer. So we'll get two layers of lipids with the heads on the outside where the water is, and on the inside where the water is, and then the tails kind of going onto the inside of the layer, forming a kind of non-polar region in the middle. We call this the phospholipid bilayer. It is where we think cells came from, this spontaneous nature of soap molecules to form these micelles and bilayers. And these are the makeup of cell membranes and the entire endomembrane system. The last group of lipids we should talk about are the steroids. Now, steroids are a special class of lipids, of fats, because structurally they are so different from the triacylglycerides we've been talking about with the glycerol and the fatty acid basics. And these are 4 carbon rings that are all fused together. And then attached to those are going to be different functional groups in different places, and where and what those functional groups are is going to change the shape of the steroid and contribute to its functionality. The classic example here is cholesterol, and here you can see the 4 carbon rings here in the middle, and then attached to them are, here's a methyl group, and here's another methyl group, and down here is a hydroxyl group. And if you were saying to yourself, well, this doesn't look like a lipid at all, why do they call these lipids? Well, this whole region down here being all hydrocarbon is, in this case, all hydrocarbon, but in most of them it's going to be largely hydrocarbon in nature, and therefore nonpolar and lipophilic, hydrophobic. But this end down here with the hydroxyl group, that's very polar and likes to get into the water. And so I have this amphipathic compound that has the hydrophilic part and it has the hydrophobic part. So it's going to line up just like the phospholipids are with the in this case, the hydroxyl group towards the water, and then this big clunky, the rest of the big clunky molecule here down in with the fatty acid chains. And that's kind of important when these guys do make up an important part of animal cell membranes. They are also a precursor. We do use cholesterol to make our sex hormones from. They are important enough that our bodies make them. Some of us are better at making cholesterol than others. And if you're like me and lucky enough to have a gene that, or genes that allow you to make lots of cholesterol, thanks mom, then you have to be careful, and those can, high levels can contribute to cardiovascular disease. But the main function here of the cholesterol that I've been talking about is to get in there in the cell membrane and kind of get in between those fatty acid legs and keep them from nesting together too close to one another, and keep that cell membrane kind of fluid and flowing, so that, remember, there's no bonds in between each individual lipid. They're just held in kind of position the same way a bubble is held together, just by the hydrophilic, hydrophobic interactions of the amphipathic molecules and the water that they are surrounded in. Sex hormones are good examples of steroidal hormones. Those are hormones that are based on cholesterol, and a good example of the importance of the functional group and the influence in the location and type of functional groups on the overall functionality of the steroid itself. Looking at cortisol and aldosterol. Cortisol is a stress hormone that influences blood flow and through capillary dilation, and like I said, it's a stress hormone, fight or flight responses, where aldosterone, certainly important, is involved in maintaining proper electrolyte balances through kidney function, through renal function. And really the big difference between the two, when you look closely, is just shifting that hydroxyl group over, or taking it out and putting a little carbonyl action on there, and you end up with poof, aldosterone. The rest of the molecule is really exactly the same. When we look at the sex hormones themselves, testosterone and estradiol, which are the male androgen and the female estrogen, respectively, really the big difference between the two, the only difference between the two, is that carbonyl group in the testosterone and the hydroxyl group in the estradiol. A good example of, the difference between the two. So there are several different functional groups that you're going to see hanging off of these cholesterol-based hormones and other compounds that are steroids. Well, that is certainly enough about macromolecules for now. Thanks for sticking it out and holding on through this extended video. We covered a lot of ground. Again, I'm Mr. Ulrich. Thanks for checking in. See you in class.